I will be discussing the recent publication, Candidate Genes Associated with Survival Following Highly Pathogenic Avian Influenza Infection in Chickens. This was published in the International Journal of Molecular Sciences, and it's a joint project between scientists from Highline International, the Roslin Institute, Iowa State University, and University of Warsaw in Poland. Recently, high-path avian influenza has been a great concern for the poultry industry. In 2015, there was a severe outbreak in Iowa that resulted in the culling of over 50 million chickens. This high-path AI was very devastating to the industry. And we were able to get some samples of birds that had survived for over 4 weeks in a high-path infected facility. This is a rare opportunity for us. Most birds are either they die or they are culled immediately. But due to delays in getting the houses depopulated, we were able to get a very small number of samples from the survivors. I also could get some samples from individuals that were from age and genetics matched flocks that were not infected. So those would be our control samples. And then, of course, we had the survivors. So the question that we had was, how is the genetics? Are there any differences in the genetics between the birds that survived and a normal flock that would have, where most of the birds would have died? And so we actually obtained, got the samples and obtained genetic sequences from the samples and then compared the DNA from the survivors versus those of the control birds. And what we found in the sequences was that there were several differences, actually 9 different chromosomes where the DNA was significantly different between the survivors and the uninfected control birds. So that was previous work we had done had shown that there was indeed a genetic influence on whether they could survive or not. But now we knew specifically where some of these genetic regions were. Now, the simplest solution for us would have been a single gene, and we could say, oh, yes, that gene is important for survival. But that was not the case. And what we actually found were 9 different chromosomes where there were genetic differences. So now what that tells us is, unfortunately, it's not a single gene that's responsible for whether these birds survived or not, but actually multiple genes in many different regions. So that actually has consequences for future studies. It means it's not going to be a simple thing just to select a specific gene and its variant, and then we'll end up with birds that are resistant to AI. It means that there's multiple regions that we're going to have to pay attention to. The other thing is that it tells us is that if we wanted to do any genetic changes to improve resistance to avian influenza, we'd have to do multiple genetic changes. So people are talking about using CRISPR and making a specific change, though we're not doing any CRISPR right now. We are focusing on the naturally occurring variation that's in the chickens. But if anybody were to do CRISPR, it would involve multiple changes, multiple edits that would need to be done. So what we did then, we focused on these 9 regions in the chicken DNA to see what are the genes that are present in those regions. And what we found was that that many of the genes in those 9 regions are known by studies done with humans and mice on equivalent genes, that they are associated with viral resistance in other species. So that was encouraging. That's confirming what we had found, that these regions are indeed important for virus resistance because they're found that way in other species. So The next question that we asked is, okay, fine. So these were commercial chickens where we collected these samples from. So they were actually birds out in the field in commercial production, egg production houses that survived. And commercial chickens are made by crosses of multiple elite lines. Now, as a breeding company, we work by improving the genetics in the elite lines, which then gets passed down to the progeny, the commercial birds. So the question that we wanted to ask was, is the variation that we see in the genes in these commercial birds also in the elite lines? It needs to be variable in the elite lines in order for us to do any selection. So we needed to check that, and that was also part of that study. And yes, indeed, the variation in those genes that we had identified as being different between survivors and the controls, the commercial birds were indeed also present in the elite lines. So that's very, very optimistic. So we could indeed manipulate the frequency of the favorable variance in the elite lines if we chose to do so. So then this kind of leads towards what future studies would we be looking at? First of all, this was a significant study. We did a lot of work, but we only had a limited number of samples. And so what we need to do is obtain more samples and repeat and see if we get the exact same regions. This would sort of tell us if it's truly are genetic differences or if it's just statistical anomalies, though we certainly did enough analyses to decrease the likelihood that it's just a sampling situation. So we need to get more samples to confirm the results. The other thing that we would like to do is, okay, these genes are in the chicken, their variation in the genes are segregating, but what we don't know is what is the impact on the variation in the genes on various production traits. It may be fine that we could say, oh, if we select for this gene, there'll be no avian influenza anymore. But if it has an impact on egg production or feed efficiency or any of those other traits, or if it makes the birds more susceptible to another disease, then that's not something that would be a favorable thing for us to select for. So the next thing that we need to do is determine the impact of these genes on other production traits, and that is currently in progress. And then the third thing is that it was very optimistic, hopeful that the genes that were identified in these regions have related functions to viral resistance, including avian influenza. But we need to confirm that. And doing high-path AI studies with live birds is very expensive. But we could do similar studies using chicken cell lines to see, well, does it impact variation in these genes, impact the cell line's ability to survive an avian influenza infection in tissue culture. Those kinds of studies can be done a lot quicker, easier, cheaper than in birds. Also, of course, cell lines are fairly easy to manipulate the genome, and so we could actually make changes in the cell line and see if this particular change does indeed impact how well the virus replicates or how well it transfers from one cell to the other. So those are the possible future studies that we would do. As a breeding company, ultimately, we'd like to be able to produce birds that were 100% resistant. You know, they don't get the virus, they don't replicate it, they don't spread it to anybody. Whether we can actually do that or not, that's a question that can really only be answered by the biology. You know, that doesn't always, just because we want to do something doesn't always mean that we can make it happen that way. For now, we're trying to understand what goes on. What are the differences? Why did some of those birds survive? Now, we had only limited access to the birds, and of course they were immediately destroyed once we obtained samples. Whether they were continuing to be producing the virus or being infective, we have absolutely no idea. Doing studies like that are really expensive. because you have to use special facilities because it's high path AI, it's transmissible to humans and other species. So a lot of precautions have to be taken to do any of those further studies, which is why I think some of the cell culture studies are worthwhile doing. So can we do it or not? I don't know. But let's get some information as much as we can about the virus and how the birds respond and the infection and everything else, so that we can then start to formulate some scientific-based questions and ask if we can, what exactly can we do with this? Maybe in the long run, a vaccination is the best thing that we can do. Don't really know yet.